and I can say a few words about you who don't know you yet. So I got to know you in your time at ETH in Zurich, where you were trained as an engineer. I think in my bubble of fMRI, you are mostly known for your advanced acquisition tools in fMRI. I know you did some kind of physiological noise stuff with Casper, Lars Casper before, but also I think specifically in the high resolution field, you're known for kind of the spirals you did with Lars, or the 3D EPI, also 3D EPI with Vaso layer fMRI that you did with a couple of students. I think at the time of when you presented your POP EPI, that was the highest resolution ever recorded in humans isotropic. The POP EPI was funny. But also when multiband was hot and new, you combine it with multi-echo. You are a proponent in our field of with respect to open science, and since a few years you have been working with Jonathan here in Boston, but also now in Stanford for a year, with the overall aim to capture smaller and smaller blood vessels with advanced acquisition, but also analysis tools that you will talk about today. And you can see the title, Towards Quantitative Analysis of Vascular Characteristics in the Human Brain. Please.
Yeah, thank you, Renzo. It was really lovely. So welcome, everyone. And today I want to talk about brain vasculature. And before we get started, I wanted to tell you a little bit about why the title is the title it is. So why quantitative? It allows us to compare across studies, participants, species. And usually the literature, if you look at vasculature, is very, very qualitative. It's images. So that's something we want to change. It then also allows to build theoretical models, and which can also then, be used in computational fluid dynamics or bolt simulations. And it also allows statistical tests, although there's a very fun exception to that rule. But in general, allows us to apply the modern tools that we have nowadays to that type of data. And then why vasculature? So vasculature plays a significant role in brain diseases like stroke, Alzheimer's, neurodegeneration. The vasculature obviously has this prominent role in metabolism and thereby also mirrors neuronal architecture. And it also has a mechanical function. For example, it helps in CSF clearance, and which circles back to neurodegeneration. And I nearly forgot about this one when preparing the slides. It is, of course, super, super important if we want to do high resolution fMRI, because in the end, we do measure changes in blood oxygenation. This blood rests in the vasculature, so helping to understand vascular anatomy can help us to pinpoint where the signal or where the fMRI signal is coming from. And the last question is, why humans? Humans are not big mice, and although we have a lot of really, really beautiful data in mice, they cannot always be applied directly to humans. For example, the ratio of diving arterioles and draining veins is different in humans. So we have more arterioles. We have cortical folding, which mice simply don't have. So that, changes quite a bit how the macron vasculature looks like. And so that's basically the reason why I think we're, there's a gap in the literature that we want to fill. We can also look at, vascular anatomy across scales. We can look at the macrovasculature, for example, with, you're all familiar with the drawings by Duvanoy. We can also look at the mesoscale, which is important for fMRI. And then we can also look at the microscale, which I won't touch up on today, although there's also a lot of interesting studies out there. Okay, and so when I say quantitative, what do I mean with that? What do we want to measure? And there's some, very, very straightforward things like the diameter, length, type, which is not always that easy. And it's actually proving more and more challenging in our work, but also in like histology. It's not always easy to distinguish arteries or arterioles and venules. If we have like diameter and length, then we can compute density, which can be expressed as like vessel volume per tissue volume. So this is basically so the blood volume. Sometimes people also report vessel length per tissue volume, and you can also, of course, compute the vessel number per tissue volume. Orientation. So it's a vessel's kind of like a cylinder. It kind of like has a orientation towards something or away something. Tortuosity is probably more relevant for clinical application. Then the location. So is it like intercortical? Is it in the leptomeninges? Is this pile? But you can also think about location. Where on the surface is it? Is it in the sulcus? Is it in the gyrus? And also, since a lot of our work is in vivo, we can also ask the question where, within the functional organization of the human brain is that vessel located and what area does it drain? So this is kind of like mostly anatomy, but we can, since the vessels form a network, we can also look at it from a topological perspective. So we can look at the connectivity, the node degree. So if you're not familiar with that term, it basically tells you how many connections a node had. So if you imagine these are kind of like vessel segments, then basically this is, the end of the vessel. And of course, the brain vasculature is a fully kind of connected system. So vessels don't end, they just become, something else. Or arterioles become capillaries, which are connected to vein. So this is a kind of like fully connected system. But of course, in our imaging, eventually we don't have the resolution to resolve that. So that's why we have kind of like ends in our graph. And this is kind of like the most typical formation where you have like one vessel that splits into two, called the bifurcation, which is probably the most predominant feature how vessels are organized. Although like technically vessels are not a tree, they since they're kind of like a fully connected network, so they have loops, it still can be helpful to think about them in terms of branching order, especially if you look at the macro scale. So you can see you basically start at the end, and then whenever that connects to like an upstream vessel, then this Strahler number increases, and it just tells you to like kind of like the hierarchy of the system. And you can also look at vascular territories. So this is again starts at from the clinical perspective, where people just look at the major supplying arteries like the posterior cerebral artery, but you can kind of like obviously go up or down in the hierarchy and look at what is the vascular territory of. Are they too loud? Can you hear them? No, okay. We can kind of like go down basically that tree and last, okay, this is obviously the vascular territory of the kind of like big branch, but what's the vascular territory of the smaller ones, and down to kind of like individual arterioles and ask how big is basically the capillary bed that they supply. So yeah, that's a bit the introduction. And as I said, I'm going to talk about brain anatomy at the macro scale and at the meso scale. And so what do we have? I did a quick search in the literature. There is a lot of really, really old literature that looked at brain vascular anatomy, but there's not too much new stuff around, unfortunately. So this is kind of like Ivana, as you all know, and about the macro scale, I couldn't really find any concrete numbers. What he says is that like arteries usually are in the sulcus, whereas veins kind of like lie on top of the gyrus. That's kind of like it. And Then there is another like interesting book again by Sikla, also pretty old, but they try to be like really, really precise. So they built this apparatus and where they can like try to, you know, like place a participant's head inside and always kind of like the same way and following that kind of like ACPC convention. And they have kind of like a lot of image, but again, you know, just images showing vascular anatomy. And I'm not sure what they did, but I found it really interesting because we're an FMI kind of like audience. They did, so these were epilepsy patients, and I wanted to localize where the epileptic foci are. So they did kind of like some stimulation, motor stimulation, and this is basically their, you know, SPM, statistical map of like the, you know, brain response to that electrical stimulation. So if you know more about this study, I would like to hear more. They also reference a paper in French, which is not my forte. And then kind of like the most, you know, kind of like classical work that probably most of you are familiar with, again, fairly old. It's the circle of Willis, which, you know, basically just describes how basically the blood supply coming through the the internal carotid and the basilar artery is kind of like distributed across the brain. But also, if you look at you know, variability across people, then you can see that this is not even the majority. So around only 30% of the population actually have this canonical circle of Willis. A large portion of the population has basically either hypoplasia or aplasia, so basically either really thin or missing communicating artery, or very often also they're just completely missing or very, very thin. So that shows kind of like another problem with you know, the literature that we have. It's very often n equals 1. So that doesn't, but vasculature is very, very variable. So it doesn't tell us a lot about you know, what our participants are doing. Okay, so far the literature is very often just descriptive, which images or text. They're difficult to like access or incorporate into modern neuroimaging studies, and it does not account for individual variability. Although there's a paper by Mike Bernier that actually looked at that. Okay, and I wanted to basically show you 2 of our studies where we're trying to target that. One of the, I mean, time of flight angiography is nothing new, but we were really trying to push it to the highest resolution that we could, so we get kind of like a more complete picture of the kind of like cortical arterial vasculature. And then I also want to introduce you to method rules, which we developed to basically do the segmentation of this data, because the segmentation is basically the first step to then extract diameter, length, and all these other measures, connectivity. Okay, so those of you who are not familiar with time of flight, the idea is basically that you have static tissue, which has a pretty low magnetization or image intensity because it has experienced quite a number of RF pulses. And you have inflowing blood spins, which have a high image intensity. You also have only seen a few RF pulses. And this is basically the classical kind of like steady state curve. And the number of RF pulses depends on the blood delivery time. And to characterize basically the contrast between the two, we compute something called the flow-related enhancement, which is basically the difference relative to the tissue. And another thing, since we were targeting these like really small vessels to get a more complete picture, was to look at partial volume effects. So we did a very, very simple partial volume model to better understand what happens if, you know, the vessel is basically smaller than the voxel, they're of similar size, or the vessel is bigger than the voxel. And at first, we ran some simulations where we looked at, okay, what's the effect of blood delivery time? So how long it takes the blood to reach a certain, branch, and what's the effect of voxel size? And as we all expect, and it's often described in literature, the longer it takes the blood to go somewhere, the more difference, the more FRE you basically lose. What hasn't been appreciated as much is that also, if your voxels are too large, you can lose a lot of the signal contrast. So it's really, really crucial that basically your resolution is at the size of the vessels you are targeting. And we have determined that, or checked that also empirically, where we basically slowly increased the spatial resolution. And here you see the flow-related enhancement, and basically the tiny small vessel and that very big vessel. And you can see that, if your vessel is already large and resolved, there's no point in going to higher resolution. But basically, if your vessel is, fairly invisible, then indeed increasing the resolution also increases the flow-related enhancement. And putting all of that together, we acquired as high resolution images as we could at 160 micrometer isotropic resolution. One of the challenges, obviously, is the very, very long acquisition time. So one slab takes 11 minutes, and this is a combination of 6 slabs. We have then asked, okay, is there like a physical limit to where we can go? And we asked Henrik Matan and Oliver Speck, because they have a prospective motion correction system, if they could help us to increase the resolution even further. And here we went to 140 micrometer, which is, again, a 33% reduction in voxel volume compared to what we had before, and is about a 90% reduction of what is clinically used. However, because the voxels are now so tiny, the acquisition time is really, really long. So one slab takes about 20 minutes. This is 3 slab, which, cover about 2 centimeters. So it's very, very slow going if we want an image like that. So to you might think that, oh, you're using higher voxels and you see smaller features. Ta-da, what a surprise. This actually opposes early theoretical and empirical studies. So in the past, people thought that larger voxel sizes are better, and I'll show you in a second why. And another thing that people very often said in literature is that we can't see small vessels because the blood flow is too slow. And again, we have basically shown that this is not the case. The voxel size is the problem, not the speed of the blood. Which was also, as you can see, a lot of study basically had that explanation in their discussion that we can't see small vessels because blood flow is too slow. And the problem, why people think that larger voxels are better, is because classically people just look at the absolute flow-related enhancement. So they just look at the difference between the magnetization in the blood and the magnetization in tissue, but not at the ratio as we have done. And if you do that, so this is basically our plot, as you have seen before. But if you only look at the difference between magnetization in blood, magnetization in tissue, then indeed, the highest number you get for the biggest voxels. And I think that's just a difference in perspective from, physics point of view, where you look at absolute signal differences, and more of a segmentation point of view, where all you really care about, can you differentiate in your image between blood, voxels filled with blood and, tissue voxels. Okay, so now we are where everyone is, we have an image. And so the next step is, how can we actually make some numbers out of that? And we developed Vessel Boost to automatically cement.
Is that a quick question?
Yes.
Sorry. Do you know what a upper practical limit of the slowest flow velocity that you get? Just I'm curious what
I'm not sure because you can always kind of like I took the plots out, but you can always kind of like optimize your TR and your flip angle to a specific delivery time. So, I mean, a practical limit is probably where you are because the acquisitions take really, really long. But I would say kind of like a theoretical limit. I'll also it also I think that is also something people don't think about. It is not how slow the blood is flowing. It is how long it takes it to get where it is. Right. I'll later show some 2D images where we look in arterioles. And there, arguably the blood is very slow, but because it's 2D, it still works. So yeah, it really depends on kind of like how you image, where you image, and
well, at some point, flow velocity will be on the order of like, the small amounts of motion. And so that seems like that's would be the determinant in that sense. I'm just wondering if you ever thought of what number that would be.
I'm always trying to tell people to not worry about it.
Okay. All right.
Yes. But yeah, I'll keep thinking about it. The other issue is that we actually don't really know how fast blood is flowing in these vessels. It's not that easy to measure, and we know it in mice, obviously. But again, human data is very, very scarce. Okay, so to continue, vessel boost. I hope some of you have heard it. If not, please check it out. So the task was basically to segment these images, and let's see how well this works on Zoom. But tell me which of these statements is true. First, high-resolution MRA can be manually segmented, or classical segmentation tools can deal with the increased noise level, or one scan is sufficient training data for machine learning. So if you wanna, do I see the chat? Maybe not, but if you wanna just put up your fingers, 1, 2, 3, or just say something. So which one of these is true? Okay, Renzo, say something. What do you think? 3. Okay. Okay. Renzo Sheehan. Okay, we have something in the chat as well. 3, 1. Yes. Okay. You already know what I'm talking about. So we tried. A colleague of mine said, Saskia, I'm doing segmentations. I do that for you. Like just a couple, like a small slab, but I give you some ground truth data. He got carpal tunnel and had to collect stuff. So manually segmenting this data is no longer feasible. Obviously, classical segmentation tools, these data are really, really noisy. They just struggle a lot with it. And interestingly, we can train on the data we want to segment. And this really, really helps us. Here way again. So this is basically where we started, or where chronologically we started with Booster, where the idea is you do some pre-processing, and then you basically give an input plus a segmentation you have somehow obtained. We do some data segmentation, and then we get an output and plus some post-processing. So this is kind of like the simple processing pipeline for Booster. And the cool thing is that you can train on the same data set. So, because, not many people will do this type of acquisition. So we are not worried about generalizability or anything. We just want to segment this one image as best as we can. And what we realize is you can take that original image, do just a simple threshold, nothing fancy, and then boom, the machine learning basically fills in the gaps that you haven't given it in the training data. So that's kind of like super, super useful for us because we don't have ground truth, we don't have a lot of training data. So that helps us to say, okay, I have this one very specific image and I want to somehow segment it. And obviously this, if you have a different contrast or a different acquisition, then, as long as you can give the machine learning model some, starting point, this should hopefully work. And time has moved on and other people also got interested in vascular segmentations. And we took part in the Smile-Uhura challenge. I don't know if you've heard, but the idea was basically to build a general segmentation network. And they provided some training data for us at 300 micrometer resolution. So we took part and very happily realized that the network that was trained at 300 micrometer resolution also does really, really well on our 160 and Hendrix 150 micrometer data. So now we also have basically a module where you can just, take, okay, these are, fairly similar time of flight data. You give it to the model and it just like predicts it. You don't have to necessarily train something new if the data is similar to what we have been using. And we have a segmentation. So let's do some analysis. First step is to smooth the volume. Then next step is to extract the center line. Now we have diameters. We have the branch points. Get the endpoints. That's for the topology. And ta-da! Problem solved. And we get also all of our numbers out there. Now, what is the diameter of kind of like each individual vessel segment? There's only one problem. This is total rubbish, unfortunately. So this does not work at all. You can see that, there are way, way, way too many brown points all over here that are not real. So although we have these, very, very high resolution images, this actually poses quite a challenge for us to extract the center line and then turn into a graph. And this is ongoing work. Unfortunately, we don't have a solution for that yet. So in summary, I hope I could show you that in vivo vascular MRI and, in this example, time of flight, provides us just digital data in 3D, which may sound a bit silly, but if you look at the literature, it's just, big books with many, many, many images. This really, really helps us. This is, the same tool, obviously, that is used in many neuroimaging studies. But we still face very long scan times, and obviously the analysis tools for that kind of data are still in their infancy. But we can also look at like larger populations more easily than what you could do with ex vivo histology, for example. Okay, so let's take one step further and let's look at even smaller vessels. Let's look at the mesoscale. So I took that picture here from Farouk Gulban, just to make a little bit of point where, I see the difference between mesoscale and macroscale, at least for the, I think there's coffee. No, there's no coffee. At least for, vasculature. So mesoscale for me is basically starting to look at intercortical vessels, including peel stuff as well when they are small, but really targeting these intercortical vessels as well. And if you're not familiar, so the leptomeningeal vessels, they're basically the vessels that lie between the two thin leptomeningles, so the pia mater and the rachis. And we have actually a bit more data here. So this is again Duvanois, and he took out his ruler to do some measurements for us. This is still on the peel surface, but you can see, the numbers are getting very, very small. Not something we usually resolve. So the central artery is basically, still around 300 microns, but then it goes down to 200. And arteries that kind of like, before they dive into the cortex and become intracortical arteries, they're below 50 micrometer diameter. And anastomosis, that's something people ask us very often about. But the sad truth is they usually form well below 100 micrometer diameter, and this is still too small for us to see at the moment. This is another study, sorry. More recent, and they built also a corrosion cask, and they looked at the branching pattern of the arterial tree using just manual dissections, but also 70 and 9.40 MRI and CT. And we have some numbers again. So they looked at like radii, length, the asymmetry ratio between different branches. So is one branch, are they kind of like split into the same diameter, or is one branch smaller than the other? You can see that usually like smaller branches branch off a bigger branch. And then I looked at, for example, length to radius. So classically, vessels are much longer than they are thick. And tapering, so that means is basically the proximal and distal end. What's the kind of like relationship or the ratio between their radii? And we see that our vessels actually don't taper. This is like another study that tells us a little bit about density. So not quite in humans, but in monkeys, so fairly close. And we can see as we kind of like go up the, like evolution, that the density of arteries or arterioles that kind of like dive into the cortex like readily increases. And this is basically what Duano had to say to that. So this is kind of like this famous venous unit in humans, where the idea is you have one vein that kind of like, drains a certain area of the brain, and that area, or of the cortex, and that area is supplied by several arteries. So again, this is very different in mice, where you basically have more veins than arteries. So here we have more arteries than veins. So yeah, we have a bit more understanding, or a bit more quantification available, although this is, still pretty general. Just, in general, the vasculature in a human brain looks like this. And I wanted to show you one of our projects where we try to make that a little bit more specific. So the story is basically Kevin Sitek approached us at some point and said, hey, I have to school data from an ex vivo brainstem specimen. Could you help and segment us? So we said, yes, let's give this a try. So he's basically interested in the midbrain, where a lot of the auditory nuclei sit. So we said, let's give it a try if we can, segment this. And this is for reference, Duvernoy's view of the like collicular region, which are like sitting at the back here. And if we compare ourselves to, like histological data, then we can see, so this is 7 Tesla T2 star weighted MRI of that specimen acquired at 50 micrometer resolution. And we can see at that level, obviously, we see a lot of very, very beautiful detail. And so how can we segment that? As you can see, the intensity profile is a bit more varied than in the time of flight data. So what we basically did to get rid of like the contrast in the tissue is to really crank up the bias field correction and like remove all of that. So we're just left up with something that's background or vessel. And then we applied basically the same trick again, a little bit differently this time. So we used our network that was trained on the time of flight images, and then had it predict on the T2 star weighted data, and used that prediction then to like retrain the deep learning model using the original weights as a starting point. And that like together gave us a pretty nice prediction of the vasculature, or pretty nice segmentation of the vasculature in that specimen. And now again, we can once you have a segmentation, you can start to have some fun. So one of the things we looked at is what's the orientation of these vessels in the midbrain. So this is basically just a section taking out. Obviously, in the cortex, we know, you have this radial orientation where like arterioles, and then you can point, perpendicular to the cortical surface. So our question is, can we find, a similar mechanism in the brainstem, or is it all just random oriented? Also a little bit quick and dirty anatomy. So basically the two areas we are looking at, the superior colliculus, so number 7 here, the inferior colliculus, so number 8, it's a little bit lower, and the periaqueductal gray, which is basically the gray matter that is around the aqueduct here in the middle. And in our orientation analysis, we basically saw that within the PAG, there's no preferred orientation, which kind of makes sense because the vessels have to somehow get around this aqueduct in the middle. Whereas in the superior and inferior colliculus, there's indeed a preferred orientation, which is of course good for us to know if you do fMRI, and okay, at least to some degree, your intensity of the Bolt signal is modulated by the relative angle to the main magnetic field. And again, we can get out numbers like length, diameter, or radius in this case. And we also looked at node degree. And if you remember, I first told you that actually we should either just have a degree of 1, which is an endpoint, or a degree of 3, which is a bifurcation. You can see that we have these higher node degrees. And on the one hand side, that might mean something very, very exciting, but I think at the moment it rather means that the graphing still needs more refinement. So this is probably still more of an error signal for us to say, okay, although the segmentation looks pretty good, the graphing needs more work, and we are working on that. So there was one of the contributions we're trying to make, or provide more numbers, make things more quantitative. Then we have seen that for the macro scale was pretty difficult to incorporate. Not even if you knew the vasculature, what would you do with it in your neuroimaging study? However, at the mesoscale, there are quite a few simulations that basically look at the effect of draining veins. And we have also done one of these studies, and also not only looking at Bolt, but also looking at vaso. And basically the idea was you can assume a certain, arrangement of the draining veins and or feeding arteries, draining veins, some baseline CBV, so this is the capillary network. And then you basically build a forward model that tells you, okay, from that example voxel, your vaso signal or your Bolt signal would like this across cortical depths. And what we tried is basically to fit the data that we've acquired, and basically the 3 parameter was the change in diameter in venules, ICAs, and ICVs. And what we found is basically that the majority of volume change is in arterioles and capillaries, and there's very, very, very little volume change within these draining arteries. Obviously, this is just a simulation study, but it shows we can, to some degree, use a little bit the information that we have about the vasculature to inform our interpretation of the fMRI data. And another problem, obviously, if you do ex vivo stuff, it's very, very difficult to account for individual variability. So I want to show you a study in not humans, but non-human primates, where we look at intracortical arteries. So this is something we have not achieved in vivo in humans, but at least again, is theoretically possible. And once you can do something in vivo, it's much, much easier to do it across many people. So that actually uses the standard 7 Tesla MRI. So no fancy setup in that sense. But the trick was obviously the high in-plane resolution. So this is now just a 2D slice. And with kind of like optimized TR and flip angle. And the trick was to really, really, really increase the spatial resolution to the level that we can, resolve these very, very small vessels. And the acquisition time for these images is around 20 minutes. So again, fairly long, but it shows that at least from a biological perspective, it is definitely feasible. And one of the key features was basically to, place that one slice such that the arterials would, perpendicular flow through it. So obviously, if they were parallel, then we would lose all the contrast. And this is the result. And to make or convince ourselves that this is not just noise, we did a test-retest check. So basically, these are 2 independent acquisitions. And you can see that these white spots, although they slightly move a little bit, the overall configuration stays the same. So these are most likely intracortical arterioles imaged in vivo for the first time, I would say. And the nice thing is, if you plan your slice really well, you can even look at different cortical areas, and then you can, start to think about differences in vessel density and stuff like that. So although it might be, which we would all prefer like a full whole brain 3D image, this can be quite valuable if you can, ask a question in a way that you can have this kind of like 2D slice. And kind of like another study or project we're working on, and where you've already seen also quite a lot of the images, is of the imaging intercortical veins. Although I slowly come to believe that we're actually seeing, at least on the macro scale, a lot of the arteries as well. But this is basically the work from Farouk, where he's basically optimized the skip-KIP segmented 3D EPI from Rüdiger Sternberg to give him kind of like really, really high or fairly high spatial resolution. And the nice thing is, because it is T2 star weighted, we have zooming effects. So everything appears a little bit bigger than it really is. So even at 350 micrometer resolution, you can see intracortical vessels. And again, here, so these are basically the ones that we are targeting for. And the great thing is that obviously you have the whole 3D volume, so you can get the whole picture. And we started with segmenting these, still needs a little bit of fine tuning, but you get obviously the gist where this is going. And yeah, so to kind of like wrap up, these are basically kind of like the points I made before about kind of like the macrovasculature and the mesoscale, kind of like the image or the challenges that we are facing, that very often they're, images or text. But I think that in vivo and ex vivo MRI can, start to provide us with quantitative 3-dimensional digital data. Obviously, you can also do a lot of histology, but especially if you want to like get, not just the intracortical vasculature, but also collective vasculature on top of the cortex, that gets very, very challenging. And ignoring like the acquisition time, these high resolution acquisitions, and also now segmentations, will hopefully eventually allow us to integrate that into neuroimaging workflows. As you know, Farouk scans are pretty fast, so that's probably something everyone can acquire. And as I said, I feel that we're actually seeing at least the large arteries in there as well. So now again, it's more a question of differentiating between arteries and vein in these images. And if we are quick enough in our acquisition, we could even acquire subject-specific vascular maps, which I think in the end is probably the way we have to do it, just because of the huge variability between people in their vasculature. Although I think it would be, insightful to have like a Statler, an atlas, or like something that tells us the variation across people. You then can't go back and say, oh, what's the vasculature of my specific participant. And with that, I want to thank you, all the people who contributed to this work, and please let me know if you have questions.
Thanks a lot, Saskia. Those were very nice images with nice segmentations as well. Thanks.
We are trying our best.
I see Peter unmuted himself already. Do you want to ask something?
Oh, who?
Oh, sorry, you were unmuted. Then
yeah. I did have a question.
Okay.
Yes.
No, this is really beautiful work. And your point at the very end, trying to look at individual variability. Yeah, I think it's really important. Obviously, if you want to know where the big vessels are and look around them or understand them. But I was wondering, just a more general question of, do you think that there's individual variability that's anything but just random? I mean, for instance, it seems that the variability that, the smaller the vessel gets, there might be more homogeneous distribution. But I mean, there could be actually some interesting differences. I mean, between individuals that are not random necessarily, that correspond to something.
Yeah. So I think that depends on kind of like the spatial scale you're looking at.
Yeah.
So we know that at least for capillaries, you can have some plasticity. Will we grow a second PCA? Probably not, no matter how much we use our brain. But I think what we don't know is, what drives this, the kind of like, initial layout in the beginning. Is it kind of like genetic? Is it, just a consequence of the folding pattern? So I think that's, something we don't know. And there's one study which made a very interesting argument that in people with motor neuron disease, you can basically have either a supply pattern where you have like just one artery supplying your motor cortex, or you can have where it's actually coming from two directions. And they made the argument that if you only have one, or basically the ratio of people with only one artery was higher in people with motor neuron disease. So again, that doesn't tell you, if this was just by chance, but it tells you, there might still be some information or some meaningful ideas in there. So, but yeah, I think to ultimately answer the question, we need to somehow, characterize the vasculature in digital 3D, whatever, with numbers, to then go out and ask this question.
Yeah, or even looking at some of the statistics of the vessel, radius distribution could be an early sign of, hypertension and, effects or things like that. So, yeah.
Yeah. Although I don't know if you've seen it, but depending on how you measure these statistics, they change a lot.
Yeah.
So I think I'm not sure if statistics or these kind of plots are really, really sensitive enough, or really if you need kind of like that combination of where that vessel is within the vasculature as well. But yeah, I mean, we know that arteriosclerosis, obviously thins vessels, so that's definitely there. Yeah.
Saskia.
Yes.
Very nice talk. You mentioned the importance of the resolution for the vessel segmentation, and then you later on around the brainstem show some Wyolm plots about the orientation. I was wondering if you want to go to the direction of how orientation of the Cartesian grid and the wane will affect the segmentation quality. Have you observed any in that direction?
Yeah, that's a good point. We haven't actually looked into that. I think Berkin once did kind of like actually did some Cosmos experiments where you can see that, depending on like the orientation, you can see different vessels. I guess for like everything that's pretty well resolved, that's such a problem. Also, keep in mind that although this is T2 star weighted, it is a specimen. So the contrast is not blood. At least the majority of the red blood cells should all be washed out. So the contrast is really just a hole in the tissue. So I don't know if then that makes it less susceptible to susceptibility effects as well. So at least for the specimen, it might not be such a problem.
Yeah, but for like actual in vivo,
yes,
I think there are plenty of wanes that are like oblique to all three dimensions, could effectively reduce the resolution, the resolving resolution and segmentation resolution, I would say. Like if the vein, for example, around the Sylvian fissure, it is like oblique. If your slice prescription is aligned to ACPC, it will be oblique into many dimensions. Then you will just have few voxels in every dimension. And that may change the segmentation quality.
Oh, yeah. I mean, they just kind of like, reuse 3D images or like 3D patches. And I guess, as long as kind of like the image contrast is similar, it doesn't really matter how they are oriented. Kind of like the network should, have the filters in place to find vessels no matter what the orientation is.
Okay, thank you.
Okay, thank you.
Saskia, I was wondering if you could say a few more words on how this overfitting works. It's like if I understood you correctly in your vessel boost, it's a kind of, you have two iterations, right? You have, which is both happening on the same data. So you do the kind of a thresholding and then the fitting on those data, and then you use that model on the data itself. So
yes.
So when I download it, I don't download a network. I download kind of I do training and testing.
You can do both. So you can do all three actually. So you can, especially if you don't have like time of flight images, you have some other contrast where you have, vessels and maybe a, segmentation you're already happy with, but which you would like to improve. Then you can use Booster to basically, train a new network for your data set that you want to, segment using the data and, some segmentation that might be good, but not as good as you would like it. And I mean, I don't, maybe it is overfitting other these, if you take these networks through other images that look similar, they still work pretty well. What you can also do is, and what we have done in a way is something called test time adaptation, where basically you take the network, or sorry, let's do it the other way around. With the Smilo-Hura challenge, we now also have a trained network that you can just use. So if your data looks like our data, then you don't have to like go through the process of training your own network. You can just use that, and then that works also really well. Or you can do basically a combination of the two, which is called test time adaptation, where you take our pre-trained network, predict on your data, and then take that prediction and retrain that network that we have pre-trained for you and get an even better prediction. I know this like obviously sounds a bit Munchausen style, pulling yourself out of your own hair, but it kind of like works. And obviously you need to check the results in the end, and these things also sometimes fail. But we found interesting enough that you can like really bootstrap your way through the segmentations and get more and more fine details.
Cool.
Hi, I'm new to all this. I was wondering if you and Farouk are already trying to quantify the vessels with the mesovein scan.
The important word here is trying. So we have collect started, but it's probably gonna still take us a little while. Also because, as you have seen, this is now like whole brain. So this is just a lot, and we have come up with ways how to actually visualize it, because classically what you've seen are these like minimum intensity projections where you just do a collapse across one dimension, and then you have like 1D image which you can assess and say, oh, this looks good, this looks bad. If we do that for Farouk's data, we just get a black blob, because there are just so many vessels in there that everywhere is a vessel. So we basically have to reinvent every step in that cascade of working with the startup. So yes, we are working on it. And if you have an area where you're like, I'm just interested in this small region of interest, we can probably do something about it. But yes, if you're just like, I want the whole brain, yes, then we should talk, definitely, because it's I mean, the data has a lot of contrast differences, because it is also T2 star weighted. So then having the whole thing segmented perfectly is quite challenging. But if you can tell us, oh, I'm just, I don't know, motor cortex, that's all I care about, then that's much more easier, because then we can really tailor the network to that area and don't ignore the rest. So yeah, definitely reach out.
Maybe I have another question with respect to maybe the ex vivo stuff that you showed. So I was surprised to see that in ex vivo, kind of, you have all these black spots. Or maybe this is something you clarified when you answered Burak's question. So in ex vivo, like, dead people are pale, right? The blood is not in the tissue anymore. It's somewhere else. How? It's not a susceptibility effect, or is there kind of something injected there, or how do you get black blood in ex vivo?
You're asking very difficult questions, friends. I've also been asked, I mean, this is kind of like the first time I'm working with ex vivo data. So I've been asking exactly the same question. One of the challenges is that people don't usually look at vessels. So they haven't really, thought about it too much. More like a nuisance for them in the specimen and for example, if you do if you think about like ex vivo T1 weighted imaging, you don't see any of that, right? I mean, there's some beautiful data sets out there, super high resolution, and you look at them, there's not a single vessel inside. So I think they, also tailored it a bit towards that. But I think like my understanding so far is that the majority of the blood has been washed out as good as they could. There may still be some red blood cells, left, but probably, and I mean also, like the hemoglobin deteriorated. So there's probably no hemoglobin left in the form we think about it. So my interpretation for now is that the contrast is really just holds filled, with whatever solution they put the specimen in, and that's what we are seeing. So it's really kind of like gaps in the tissue rather than blood.
Okay.
But yeah, if someone knows more about this, or I don't know, has a microscope and has looked at this, I'm curious to hear more. But yeah, I think that is the story we are sticking to at the moment. And also the question is, what else should it be?
Right. Yeah. Sure.
So it's definitely a vessel, I think. But yeah, how exactly we are seeing it, we are still trying to understand that more.
Maybe I do have one more question about this kind of subdiscipline within vessel imaging that got a lot of attention maybe 10 years ago. You mentioned Michael Bernier, but also in Stanford, I think they had this ferrum oxetol a Ferraheme injection, kind of FDA cleared, and then there was a warning again. And what's the status there? So because you would get a much more signal, right, with contrast agents, and is somebody doing that for high resolution vessel imaging, or has this stopped because of the black book warning, or what's the status in that field?
Well, I don't know about the warning, to be honest. What I can tell you is that John did some studies at the MGH, and they also acquired quite a few participants, actually. I don't know what the status is. I think, again, you're seeing even more. So the segmentation becomes even more challenging. And I think like these data have even more susceptibility artifacts, which just makes it even more challenging. So I know that the data, or at least some data, has been acquired and quite a few participants, but I think it's really now stuck at the analysis side. And which I think also shows us how important this tool development is. That we can acquire a T1 image and set it for free surfer and have like a thickness within I don't know how long fast surfer takes nowadays, maybe 20 minutes or so. But anyways, everyone and their dog can do it, and but for vessels, there's just nothing. If you go like outside the clinical realm, and even there, it's just looking at images most of the time. So I think we have more acquisition techniques than we have analysis techniques, and we really need to develop more of this tooling stuff to make something out of the data.
All right, thanks a lot. We have 5 past, so if there's no further burning question, thank you, Saskia. And
thank you.
A note for everyone, we will not meet in 2 weeks because it's very close to Christmas. We will meet again in January.